Hello, we'll continue our lecture with urine analysis. We'll talk about the kidney and the glomerular and renal tubular function, urine specimen collection, and urine analysis. Basically, the function of kidney is the functional unit of the kidney is the nephron. The function include regulation of water, electrolyte, and acid-base balance, excretion of products of proteins, nucleic acid metabolism like urea, creatinine, and uric acid. The kidney as well considered to be one of the endocrine glands producing a number of hormones are subject to control by others. AVP act to affect water balance. Aldosterone affects sodium reabsorption in the nephron. Parathyroid hormones promotes tubular reabsorption of calcium, phosphate excretion, and synthesis of the 1,25-dihydroxycalciferol, which is the active form of vitamin D. Renin is made by the juxtaglomerular cells and they catalyze the formation of angiotensin 1 and ultimately aldosterone synthesis. The renal function is always discussed in terms of glomerular and the tubular function. So if you look closely here, you'll see the Bowman capsule, which is the glomerulus, which contains the glomerulus capillaries, and this is lead to the proximal tubules where reabsorption occurs, then the lobe of Henle where the concentration of filtrate occurs, and then to the distal tubules where secretion occurs, and finally all of this is collected in the collecting duct for water reabsorption. If you look closely to the functions of the kidney, you can see that it can be stimulated with AVP from the posterior pituitary, parathyroid hormone from the parathyroid, and aldosterone from the adrenal cortex. Aldosterone needs to be activated by the renin secreted from the kidney, which convert angiotensin into angiotensin 1 and 2, which activate the aldosterone to affect the kidney. Also, the kidney produce erythropoietin, which help in the development of the bone marrow, as well as the active form of vitamin D for the gut. And this figure shows the endocrine links in the kidney. If you look closely to the function of the nephron, you can see the parts that do, every part have a specific function, either glomerular filtration, tubular reabsorption, or tubular secretion. And finally, all the leftovers are collected in the collecting ducts for the excretion. That's a method of urine formation. For glomerular function, the first step in urine formation is the filtration of plasma at the glomeruli. This GFR, glomerular filtration rate, is defined as the volume of plasma from which a given substance is completely cleared by the glomerular filtration per unit time. It's approximately 140 ml per minute in a healthy adult, but this varies according to the body size, and usually it is normalized to take account of this. This GFR is normally corrected to body surface area, BSA, as the unit are ml per minute per meter square. The measurement of creatinine in serum has been used as a convenient way but insensitive measure of glomerular function. The following figure shows how the relationship between the glomerular filtration rate and serum concentration, serum creatinine concentration. And you can see that glomerular filtration rate may fail considerably before serum creatinine is significantly increased. You can see the reference range of the serum creatinine, and as long as the glomerular filtration fails when the concentration is high of the serum creatinine. The normal serum creatinine doesn't necessarily mean, if we have a normal value for serum creatinine, doesn't necessarily mean all is well, because, for example, consider a person who doesn't show any symptoms, have a serum creatinine of 130. In a young woman, this might be abnormal and requires follow-up. In a muscular young man, this is expected result. In an elderly person, this may affect the physiological decline of GFR with age. So what is creatinine clearance? The simultaneous measurement of urinary excretion of creatinine by means of a timed urine collection allow estimation of how the body clear creatinine. The way this is work as follows: the amount of creatinine excreted in urine over a given period of time is the product of the volume of the urine collected, okay, in 24 hours, and the urine creatine concentration is zero. So this work out the volume of plasma that will have contained the amount of creatinine. The amount excreted is divided by the plasma concentration, and you can see from this equation how we calculate the creatinine clearance. This is the volume, actually, if we want to define, this is the volume of plasma that would have to be completely cleared of the creatinine during the time of collection in order to give the amount seen in the urine, thus known as the creatinine clearance. Although it is more sensitive than the serum creatinine in detecting reduced GFR, it is inconvenient for patient and imprecise, and has now been largely superseded by the so-called prediction equation with estimated GFR. So estimated GFR, it's considered to be a better one. The relatively poor inverse correlation between serum creatinine and GFR can be improved by taking into account of the variables like age, sex, ethnic background, and body weight. So this formula developed by Cockcroft and Gould in the 70s, and the four variable equation derived more recently from the modification of the diet in renal disease study are the most widely used of this prediction equation. Okay, so this table only for knowledge to show how you can differentiate between the two ways of measuring the creatinine. It's only for knowledge. So although these prediction equation are an improvement on serum creatinine and the creatinine clearance, they are merely estimates of GFR and should be interpreted with caution. Patient groups may have less accurate GFR because of abnormal body shape or mass, like muscle wasting or amputees. There is evidence that GFR estimated by the MDRD formula is affected by consumption of meat. Proteinuria, it's another aspect of glomerular function, is the leakage. So if we look to the renal tubular function, to ensure that the important constituents such as water, sodium, glucose, amino acids are not lost from body, tubular reabsorption must be efficient. For example, 180 liters of fluid pass into the glomerular filtrate each day, and more than 99% of this is recovered. Compared with GFR as an assessment of glomerular function, there is no easily performed test that can measure tubular function in a quantitative manner. So it's harder to measure quantitatively the renal tubular function. If the tubules and the collecting ducts are working efficiently, and if AFB is present, they will be able to reabsorb water. To assess tubular function conveniently done by determining the urine osmolality and then comparing this to the plasma. If the urine osmolality is 600 and tubular function is usually regarded as intact, so they take the measurement of the urine osmolality as a reflection for the tubular function. When the urine osmolality doesn't differ greatly from plasma, ratio around 1, the renal tubular are not reabsorbing. Renal tubular dysfunction is one of several causes of disordered water hemostasis. Going back to proteinuria, the microglobulins are filtered at the glomerular and again reabsorbed by the tubular cells. An increased concentration of this protein in urine is a sensitive indicator of a renal tubular cell damage. If you look to this table, it's also for knowledge. It shows that the causes of polyuria, the polyuria is an excessive or abnormally large production or passage of urine. So you can see how differs the urine osmolality from the plasma osmolality, which lead to polyuria. Urine pH measurement may be useful as a first step in the diagnosis of renal tubular acidosis, RTA, which typically give rise to hyperchloremic metabolic acidosis. RTA may be characterized as follows: type 1, defective hydrogen ion secretion in the distal tubule that may be inherited or acquired. Type 2, it is the capacity to reabsorb bicarbonate in the proximal tubule is reduced. Type 3, it's in pediatric variant of type 1 renal tubular acidosis. Type 4, the bicarbonate reabsorption by renal tubule is impaired as a consequence of aldosterone deficiency, aldosterone receptor defect, or drug which block aldosterone action. These slides show some highlights and the important highlights of the topic we already discussed now. And usually these slides is a kind of important slides for studying. Talking again about urine, so it's a process of cellular metabolism. That any cellular metabolism generate numerous waste products and compounds, and these are rich in nitrogen that require elimination from the bloodstream. This waste is eventually expelled in the process of urination, and of course urine is a liquid that pass through the body, outside of body from the urethra. An ultrafiltrative plasma from which glucose, amino acid, and all these compounds are essential to body metabolism are again reabsorbed. Urine carries waste product and excess water out of the body. These chemicals can be detected and analyzed by the urine analysis. Urine test checks different components of urine. So in a regular urine test may be done to find the cause and symptoms. The test can give information about your health and problems you may have. Urine has hundreds of different body waste when what you eat, everything how much you exercise, a lot of body waste. In pregnant women, amniotic fluid is very closely related to urine and can be analyzed by amniocentesis. If you look to urine, it's 96% of water, 4% dissolved salts. Half of that is urea and half of that is other compound. The other combined either inorganic or organic like creatinine and uric acid, and inorganic some trace metals and trace amount of sulfate and bicarbonate. The factors that affect urine constituents is the dietary intake, physical activity, body metabolism, endocrine function, and a lot others. This slide also for knowledge show the composition and the percentage of each component in the urine. For specimen collection, as you know, the urine analysis provide information about how the kidney are function and if waste are being properly filtered from the body. Prior to collection, the external genitalia must be cleaned with an antiseptic solution for accurate urine analysis and the proper collection technique. The first important step is the use of clean dry container. Disposable containers are preferred for by most laboratories. Samples that to be cultured must be collected in a sterile container. During the collection, the initial portion of the serum had to be escaped, while the midstream portion is collected into sterile container. In order to obtain suitable specimen for infant and small children, pediatric urine collector which are attached to the genitalia are available. Specimen collection, there is different ways, different types for collecting specimen. The clean voided midstream specimen, which can be called clean catch specimen, it is the simplest method of collecting urine, is usually the method of choice. Specimen obtained after external urethral area is washed with a liquid soap and rinsed well. Then the patient start a urinary stream. Then the patient stop it and void it into a sterile specimen container. Catheterization performed by inserting a plastic or rubber catheter through a urethra into bladder, performed aesthetically to prevent infection and is used in emergencies and for immobile patients that need long-term care. Cytosynthesis suprapubic aspiration is like performed by inserting a needle through the abdomen into the bladder. Aspect condition technique is used to prevent infection. Performed to obtain a pure urine sample or to relieve bladder pressure on an obstructed patient. For the routine urine analysis or routine UA, it consists of group of tests performed as part of physical examination. It involves macroscopic and microscopics. The types of analysis either, as we said, macroscopic, physical and chemical analysis, or microscopic under the microscope. Steps in basic urine analysis, there are 3 steps. The first one is the physical characteristics of urine are noted and recorded, and followed by a series of chemical tests is run, followed by urine sediment is examined under the microscope to identify the components of the sediments. So we here summarize, there is a first step, gross appearance of the urine, volume and color. Then we see under the microscope the cells, casts, crystals, bacteria inside the urine. Then you do the biochemistry to check the pH, osmolality, protein, urea, creatinine, and glucose. So types of urine specimen, type collection are determined by physician and depend on the test that will be performed. There are basically 4 types of urine specimen: first morning specimen, random urine specimen, fractional collection, timed collection. The composition and concentration of urine changes during the 24-hour. Urine concentration vary according to the water intake and the physical activity. If you need a truly representative sample, it is necessary to regulate the time of collection, the length of collection period, the patient dietary, the medical intake, the method of collection. Initial morning sample is preferred, particularly for protein analysis, because they are more concentrated from overnight retention in bladder. The time of analysis must be analyzed within 1 hour at room temperature or within 8 hours if it's stored from 2 to 8 degrees centigrade. If not analyzed within these time limits, severe change will occur. Sample should collected in a clean container. Urine container must be sterile if the urine to be cultured. For microscopic examination, the urine must be fresh. The first test, as we said, the physical characterization. You need a direct visual observation. The volume must be taken maximum from 750 to 2 liter per 24 hour. Must be normal fresh urine, have a color pale or dark yellow, amber, clear. The physical examination involves color, transparency, odor, volume, pH, specific gravity. For the color, the urine color is affected by fluid balance, diet, medicine, and disease. The intensity of urine correlates with the concentration. The darker the color means the concentrated sample. If it's colorless, due to reduced concentration. If it's amber yellow, urochrome, that means there is a derivative of bilirubin degradation, a pigment found in normal urine. If it's silver or milky appearance, that means there is a pus, bacteria, or epithelial cells in the urine. Reddish brown, blood in the urine. Yellow foam, bile of medication in the urine. Orange, green, blue, or red medications. Vitamin B supplements can turn urine bright yellow. For transparency, it's classified as the urine is classified as clear or turbid. Urine is normally clear. Bacteria, blood, sperm, crystal, mucus make urine cloudy, as you can see in the figure. In normal urine, the main cause of cloudiness is crystals and epithelial cells. In pathological urine, it is due to pus, blood, and bacteria. The degree of cloudiness depends on pH and dissolved salts. Turbidity may be due to presence of bacteria. Smoky appearance is seen in hematuria. Thread-like cloudiness is seen in sample full of mucus. pH as a physical characteristic. You can see that pH measure acidity or alkalinity of urine. Normal urine pH is around from 4.5 to 8. Increased acidity in urine is due to diabetes or medication. Urine sample must be fresh. Standing urine become alkaline as a result of ammonia liberation due to urea decomposition. Urine pH of 4 is strongly acidic, while 9 is strongly alkaline. Sometimes the pH of the urine is affected by certain treatment. For example, your doctor may instruct you how to keep your urine either acidic or alkaline to prevent some types of kidney stones from forming. Specific gravity. This measures the amount of substance dissolved in the urine. Also indicate how well kidney are able to adjust the amount of water in urine. Higher SG, this means more solid material is dissolved into urine. When you drink a lot of fluid, your kidney make urine with a high amount of water in it, which will have a low specific gravity, and the high specific gravity will be attained vice versa. Second type of analysis is the normal chemical constituents of urine. So, as we have said, there is organic and inorganic constituents. For the organic constituents, the other urea, uric acid, creatinine, chloride, phosphate, carbonate, ammonia, sulfate. Of course, you don't need to memorize the equation, okay, but you need to memorize the result. Like urea, there is evolution of nitrogen gas. Uric acid, there is a blue color. Creatinine, there is a red color precipitate. Okay, so these are the inorganic compound which are tested as a chemical constituent of urine, while the organic one are urea, uric acid, and creatinine. For the chemical characteristics, there is protein. Protein is a family is normally not found in the urine. Fever, hard exercise, and etc. may cause protein to be in the urine. Glucose is a type of sugar found in blood. Normally very little or no glucose in urine. When blood sugar level is very high, as in uncontrolled diabetic patient, the sugar spills into the urine. Glucose can also be found in urine when the kidney are damaged or diseased. Nitrites. Bacteria that cause UTI make an enzyme that changes uranium nitrate into nitrites. So you can test for that. Leukocyte esterase. Leukocyte esterase is the test to detect leukocytes in the urine. Ketone bodies, product of breaking down fat for energy. These are passed in the urine. Large amount of ketones in urine may mean a very serious condition, example diabetic ketoacidosis. A diet low in sugar and carbohydrates, starvation, or severe vomiting may also cause ketones to be in the urine. As for lab practice, you need to have this kind of table to record your data. So you need to collect urine in a clean container, run routine urine analysis in sample using both urine strip and the method described before for chemical analysis, record the result in the lab report of the urine analysis as this table. Thank you.